Okay, we're back. So let's get started on this particular model. All right. So the thing to understand is what is an operon? Okay. It turns out bacteria have a tendency to utilize their DNA very efficiently. Okay. They have a limited amount of space, and thus they're utilizing every bit of the DNA strand. One of the things that they have built into their system is that enzymes that are involved in the same particular biochemical pathway will sometimes be linked up together under one promoter. Thus, what will happen is you'll have maybe one or more genes linked up together on one transcript. Okay, we call these things operons. This is our first understanding of how these work in these systems. So if you look here, you have a promoter system right here, and then after that you have the LAC-Z, which encodes for the beta-galactosidase, LAC-Y, which encodes for the lactose permease, and then LAC-A, which encodes for galactoside transacetylase. All right, so these are all built behind one promoter. And if you'll notice here, the operator site sits on top of where the promoter and the first gene are actually associated with each other. So this is going to be the site of regulation that we're looking at. In addition to this LAC operon, if you remember, this particular gene has to be turned off because the cell is not utilizing lactose. If the glucose is present, this gene has to be repressed. Okay, so just upstream of this is another gene, and this is at a low concentration. It is a constitutively being expressed gene. It's a housekeeper gene, and this is what generates the repressor protein. This repressor protein is going to be generated. It's going to four of them are going to come together, creating a tetramer, and then that creates the structure necessary to bind the DNA. And what it does, it binds to this operator site and just kind of loops it up so that nothing can bind to it. Just binds it, holds it together, and it creates a loop that prevents the RNA polymerase from binding. Now remember that I said that this is not a covalent binding. It's a hydrogen bonding. It's off and on, off and on. And so it will bind, but then it'll let go. And when it lets go, that gives enough time for an RNA polymerase to come in and generate a low amount of these enzymes. Remember, those enzymes have to be present in order for the cell to respond to this suddenly new lactose environment without glucose. So this is how they keep this on there. So every once in a while that repressor protein will fall off, an RNA polymerase will come in and generate, but it's a very, very low concentration of these particular enzymes. All right? They don't want to waste a lot of energy keeping this gene on if they're not going to use it. It's just a waste of polypeptides. All right, so in a wild type system, what you have is you generate a messenger RNA off here. So once you get an expression off of this, that comes off, and now you have this long messenger RNA that has this gene here, and this gene here. And for each one of these is a start site and a stop site for translation. So a ribosome can bind here, generate the LAC-Z, stop, then bind to the start site for this one and start another translation event, stop, release the polypeptide, same thing here, and then move over, start the translation of this event. And this is what we refer to as an operon. Now we know this is how it works because we've created mutations in there, nonsense mutations that will block it and cause it to fall off, and then we don't get any of these genes being expressed. So we know this is how the system works. You have a very long transcript. We call it a polycistronic messenger RNA because it contains more than one gene, or the ability to express more than one polypeptide in this particular situation. So it's a polygenic messenger RNA or a polycistronic messenger RNA. And so we know this exists in this system. Okay, so this is the basic thing we're going to be working at. We're going to be focused in on this particular area from this point on. So that's called an operon, and as I said before, Jacob and Minot, they provided this particular operon model for the regulation of LAC genes. They provided the first model for the fact that gene expression can be controlled at the level of gene transcription, okay? So they were the ones that originated the idea that the control of enzyme levels in all cells occurs through the regulation of transcription. As a result of that, they won the 1965 Nobel Prize in Medicine. It was a big event, what they did, okay? So, an operon, as they presented it, is a cluster of genes, the expression of which are regulated together by an operator-regulator protein interaction. So it's binding to that region. Also includes the operator region itself and the promoter. So the operator region is going to be that controlling site in this particular case. All right, so here it says lac operator, okay? That's that controlling site. Notice that here's the promoter, here's the beta-galactosidase gene, and see how this sits on here. So this is where the repressor protein that comes off here, it forms into a tetramer, and then it binds to this and prevents the RNA polymerase from being able to bind to it. But remember, every once in a while it comes off, an RNA polymerase comes in, and we get a polycistronic messenger RNA come off of this. So the LAC-I gene encodes a repressor protein. As I mentioned before, it's made constitutively. It binds to the operator region in the LAC operator. Because the promoter is relatively weak, this is a very weak promoter, there are only a few repressors are present in the cell, okay? But it's enough to keep that particular gene turned off. So here you have your regulatory gene here, you have the promoter, you have the operator site, which we refer to in general terms as the controlling site, LAC-Z, LAC-Y, LAC-A. All right, so we got everything in place, three-dimensionally, or two-dimensionally in this case. So, in the absence of lactose, as I said before, they're going to keep this particular system turned off. They don't need it. They're going to be utilizing glucose, okay? So the lac operon, in the absence of lactose and in the presence of glucose, the lac operon is under a negative control. That means these repressor proteins are being generated. They form into a repressor structure, which is a tetramer, and they come up here and they bind to the operator, okay? So this is what it looks like in more of a two-dimensional shape. This is just showing a basic concept. It binds to them. You'll note that we're showing here, there's a little circle here and a little circle here. That is the area where the allolactose is going to bind here, and we're going to talk about it in a few moments, to cause this to come off. When that allolactose binds to that, that changes the shape of the repressor so that it can no longer bind to that operator region up here, and thus, where the RNA polymerase is, it can zip through. Notice how the RNA polymerase is here, but this is a roadblock. It's blocking its ability to get past that. If this comes off, boom, it takes off, and we get gene expression on this. So there is a low level of gene expression. Remember, if that just comes off on its own, you get a low level, and this is due to the fact that the repressor binds and unbinds, which allows for a low amount of it. All right. Now, let's suddenly bring in the presence of lactose without glucose. What happens there? Well, as I mentioned before, because there's a low level of gene expression of these particular genes, those enzymes are present. And so if lactose is present, it'll come into the cell by way of the permease. Okay, remember that's this guy right here. That will bring it into the cell, and then the beta-galactosidase, this one here, will start to utilize it. And as I mentioned before, because it's not always a perfect binding when it does its activity, what will happen is the lactose will be conformationally changed into another isomer, which is allolactose. Later on, if that allolactose binds to another enzyme of beta-galactosidase, it will cut it in half. So allolactose becomes available, and when it becomes available, it acts as the effector molecule, and in this case, it is an inducer. It induces the gene expression. So here we have LAC-I, which remember builds the repressor proteins. Here we have the inducer molecule, allolactose. It comes in, binds to this, and changes its shape. It can no longer bind to the operator. Also presented in another model shape here, here's the repressor protein. It shows a tetramer built in place here. So you can see there's 1, 2, 3, 4. See the little sites for the allolactose. Now the allolactose binds, it changes the shape of the DNA binding domain up here. It can no longer bind to the operator, and now we get an RNA polymerase, okay? So this is inducible control. Previously, it was under negative control, which is the basic concept people thought at the time was that most genes are under a negative control. They're turned off, these inducible genes. This is under inducible control, meaning that we can induce the gene expression. In the presence of lactose, the above beta-galactosidase, low level of expression, produces inducer molecule, allolactose, which is the inducer. Allolactose, not lactose, causes an allosteric shift in the shape of the repressor. It goes in and modifies it and causes it to change its shape. So here is a cell that has a low level of galactosidase, a low level of lactose permease. It's got the lac repressor in place that's binding to the lac operon and keeping it turned off. It has some transacetylase in there as well. Now, suddenly, lactose enters the environment. As a result of that, this permease starts pumping it in here, and as a result of that, that beta-galactosidase generates some of those allolactoses. Well, that binds to this repressor protein, lifting it off. Now, we're having more permease in the surface of the cell, in the membrane. We have more beta-galactosidase, more transacetylase. It is really utilizing the system at maximum. Now we go over here and suddenly we throw it, we put glucose back in there. If you put glucose back in here, it's going to turn off this whole system, and we'll see how this works in a moment. It'll turn off this whole system because it no longer needs to use lactose. It will use glucose preferentially over the lactose. Now you got too many proteins, well, those are going to be eliminated and you'll go back down to a low level. That's the concept. So when lactose becomes available, which is basically what I've said here, most proteins involved with lactose utilization are going to be degraded. So this pretty much 1, 2, 3, 4 fits in 1, 2, 3, 4. That's where that comes into play. So you can read through that and understand this in a little bit more detail. All right, so as I mentioned before, just to make sure you understand this concept, the lactose is out here, the lactose permease with hydrogen protons is pumped into the cell. Now when it's in the cell as lactose, beta-galactosidase will typically cut it to pieces into galactose and glucose. But a side reaction based upon this will cause it to be formed into allolactose, and that is going to be the signal to bind to the actual lac repressor and cause it to come off. Eventually that particular allolactose is going to bind again to beta-galactosidase and be broken down into a usable form. So that's just kind of reflecting the same information in there, just to see it in a better way. All right. Well, how did they figure this out? Well, they had to do a series of experiments to figure this all out. And they had to use a partial diploid that has what we call an F' plasmid. So if you remember, I talked about plasmids. These are small circular forms of DNA that exist in all bacterial cells. And one of these is involved in the conjugation. It's called the F conjugation. Well, sometimes that integrates into the actual bacterial DNA. And when it integrates, it can also come out of that bacterial DNA. And sometimes it does it in a way that it captures some of the bacterial genes. Well, they found an F plasmid that actually captured this lac operon. And they go, oh, cool. So they isolated it, made more copies of it, and they decided to use this as their system for testing this particular gene expression. And so they would introduce mutations into these F plasmids, or they would introduce mutations into bacterial cells. So they would take their modified F' plasmids, is what we call them, and that's represented in this particular drawing here. They would take these and put them into a bacterial cell, this is the E. coli segment, and compare what would happen if they have these different mutations. So in this one with the bacterial cell, basically they had a mutation. They had a mutation of lac Z, so they could quickly determine that that's the operon that was on. Whereas on the F', they had a mutation for the permease, and they could quickly assess if that operon was on. So they could tell which operon was on based upon whether they had the inducer there or not. So in this case here, no expression in the absence of the inducer. They knew that they didn't get this at all. And so when they added the allolactose, then that would cause this to be expressed, but they also created a mutation in this controlling site so that the repressor couldn't bind to this. So in the absence of the inducer, this particular system operon's off, and this particular operon system is on. All right, we're going to stop for a moment so that we can continue with this particular system.